last but definitely not least, is obviously a man who needs no introduction, but I'll introduce him anyway. So come on up, Dr. Francis Collins. We're so pleased that you're here with us tonight, and thank you so much for sharing your time. We know you have a very busy schedule, but we're so pleased that now you're stuck with us, Dr. Collins, that you made time for us. So thank you so much for being here. Okay, here's your
Well, thank you, Melissa, and thank you especially for sharing your story, especially the story of Ginny, which is an appropriate way to focus our attention on why we're all here. And thank you also to Doug and Mindy in telling us about your family, about Jake and Jordan. And we shouldn't leave out Brantley here. And I do want to say how wonderful it is to be at this occasion, especially because of the involvement of the person who just received that special award, Joe. Joe was my postdoc in a laboratory at NIH a few years ago. It wasn't that recent, but it sticks in my mind as one of those amazing experiences of having a chance to mentor somebody with enormous talent and drive and vision. But when Joe was in my lab, he was working very much on technology development, figuring out how to use microarrays to do all kinds of interesting things, even DNA sequencing. Before that was such a common thing, he was right there on the leading edge of that technology. But Joe, along the way, in his faculty position at USC, discovered this community, all of you, GFPD. And here's a guy who maybe would have taken a different track and spent most of his career doing basic science and technology development, and instead he fell in love with the opportunity, not just to do science because it was fun, but by the way, it is fun, but also because it might really make a difference for people who are really desperate for somebody to care about a rare disease that hasn't gotten nearly enough attention. And Joe found that to be such a compelling thing to spend his time on that I know, because we're regular email pals, and when Joe comes to the East Coast, he stays at my house. You all are the reason that he is most excited about what he can do scientifically right now. So when he said, would I come and be part of this, I said, yeah, just tell me where to be and what to say. And I'm glad that's all come together here this evening. So I want to bring you greetings from the whole National Institutes of Health family, because I think we are a family as well. You might think of this enormous, sprawling enterprise with its $34 billion budget and its 400,000 scientists that are supported all over the country, and about 19,000 that work right near here in Bethesda on the Intramural campus as a government institution, and I guess we are that. But we're very much an institution with a heart, and particularly in the circles that I get to travel with, a heart that involves trying to do something for families like yours, and particularly for those with rare diseases that are poised on the moment of opportunity, because we're beginning to learn so much about the cause and the diagnosis, but yet we still have a long way to go in terms of turning that into treatment. So that's what I really want to talk to you about this evening, is what does that look like, trying to cross that bridge from knowing something about basic biology of a disorder and coming up with a means of intervening that is going to be helpful. Now, I confess I don't know a lot about paroxysmal disorders, so I would begin to talk to you about that class of disorders. You have people in this room who are the world's experts, and you really do, and you've nurtured many of them. And because you have meetings of this sort where you bring together the scientists and the families whose lives depend upon progress here, you are not going to see those scientists walk away, because they've been convinced that this is what really matters. And so, again, I think this is a great example of how to do this right. So I won't talk to you about paroxysmal disorders. I'll talk instead about another rare disease that I do know something about, because it's the one that's worked on in my own research lab. And there might be some parallels here. There'll be some differences as well. But I know as you are seeking how to accelerate this process of finding therapeutic answers, it's kind of good to look around and say, okay, how has that been approached in other disorders? The one I'll tell you about doesn't have all its answers, I can tell you that. Some progress, yes, it's still a ways to go. But we learned some lessons along the way that I thought might potentially be of interest. So again, NIH is your partner in all this. We do both basic science, the top part of that mission statement, but we worry about how to speed up the process of applications. That's the bottom part of the mission statement. And we do that for common diseases and rare diseases. We do that for everything where we think there's a scientific opportunity. And we depend on the taxpayers to provide the resources, and we think it's a really good investment. And by the way, I'm happy to say the Congress of the United States, as recently as this afternoon, debating about the budget for NIH for next year, fiscal year 18, agreed that this is a good investment and basically marked up the budget from the House side to say no to that 22% cut that was proposed a little earlier, and instead to say, let's not just keep it flat, let's make it go up. And they voted for a $1.1 billion increase next year for NIH. So, responsible stewards of that opportunity for you and for all the others who depend on it. Now here is something that I think graphically maybe lays out why this is both a time of such great promise and also a time in many ways of frustration. If we look at how many disorders have had their precise molecular basis uncovered, look what's happened over the course of the last 2 or 3 decades. And why did that happen? A lot of that was the Genome Project, providing the technologies to be able to survey a DNA sample and find out, oh, there's a misspelling there, and that must be the cause of this disorder. So look at this now, 6,000 disorders for which we have that kind of molecular information. Peroxisomal disorders are in there. Now here's the not so good part. How many of them have an approved treatment? Probably about 500, that's it. So we have this huge gap between what we can diagnose and what we know how to treat. And that is the mission that I think NIH has embraced with as much energy as we can. But it's unfortunately not the case that you can come up with a solution that will apply to hundreds of these disorders at one time. Each one seems to have its own challenges, its own biology, its own need for a particular approach. There are things we can generalize, strategies, and I'll mention a couple of those. But this is hard work, and it takes dedication, it takes really good scientists, and it takes resources. So I am going to tell you about a different rare disease, and it will start with a story of individuals that I came to know that, as will often be the case when you talk to scientists working on a disorder, it starts that way. You don't start working on this condition because you read it out in a book. Oftentimes it's because you encountered a family where this disorder had occurred. What we're trying to do for this is to cross that bridge from fundamental knowledge to application of that knowledge. That's what I'm mostly going to talk about. So this was the first person who got me interested in this condition of premature aging called progeria. This is Meg Casey. She was my patient when I was a fellow in training at Yale back in the early 1980s. And she, at the time, this photograph's about 20 years old, and that was quite remarkable because very few individuals with progeria had lived beyond about age 12. And she was one spunky character, I can tell you. She could out-curse most sailors if the need arose. And despite her diminutive stature of about 3 feet tall and her bird-like voice, she got her way very often when she entangled with anybody who tried to tell her that she was a child and didn't belong where she was. And she rather single-handedly took on the little town of Milford, Connecticut, and had them make everything accessible to people of her stature. And she was my patient, and I always enjoyed Meg coming to talk to me because she would always have interesting stories to tell. But I didn't have much to offer her in terms of how to manage her disorder because it was so rare. Could find only a few cases, and there was no idea what the cause was. But I thought about someday I'd love to have a chance to try to figure this out, but there didn't seem to be a way to do it. Well, then fast forward about 16 years. And here on the front steps of my house in Rockville, not far from here, is a little boy named Sam. Sam, at this point, was about 3 years old. His mom, sitting to his left, your right, Leslie Gordon. There with my wife, Diane, and me. Leslie Gordon turned out to not only be the mother of Sam, but she was also an MD, PhD. And Sam's dad was an emergency room doc. And having had a child who had just received this diagnosis of progeria a few months before this, they were determined to do something about this. And they came to see me. And Diane and I felt like maybe there's something we could do here. I suggested some ideas to them about kinds of research that might be appropriate. Suggested they should hold a workshop, kind of like what you all are doing. See if you could get some scientists interested in this issue. And they started doing that, and pretty soon I was interested too. And pretty soon I asked a postdoc in my lab, hey, why don't you work on this and see if you can figure out the cause, even though it looks like this is a really hard problem. Well, over the course of those years, now it's been 18 years since this picture was taken, I've certainly become totally immersed in trying to understand this disorder and come up with some ways to do something for it. So what is Hutchinson-Gilford progeria syndrome? That's the full name. We're just going to call it progeria, but sometimes it's HGPS. So these children are extremely rare. It's about 1 in 8 million births. They appear normal at birth. You can see in the top right there a series of photographs taken over a period of time of a single individual as a baby looking normal, then losing hair, developing this wrinkled appearance of the skin as an extremely aged person. They have normal intellectual development, very significant growth retardation, generally not growing to more than 3.5 feet tall. They have a lot of skeletal problems, but most significantly, they have heart attacks and strokes. And they die at age 12 or 13 if not given some sort of intervention, and there was no intervention when we started this, generally of a heart attack. And this is a tragic story to sort of watch and see these kids who are in many ways precocious in terms of their intellectual development, and yet their bodies are failing them, aging at about 7 times the normal rate. So what to do? Well, we wanted to figure out the cause. By the way, it was not so easy to find individuals. You all talked about rare diseases, and we certainly have another one here. RCPP, you had only maybe 100 patients. Worldwide, because the Progeria Research Foundation has scoured the world here, perhaps as many as 300 patients at any given time, but that's about it. Which means that if you want to do research, it's a real challenge to find enough individuals to learn what we need to learn. So I want to talk to you about insights that over the course of time we've come up with this, and this is based on a little article that I wrote with Leslie Gordon, that's Sam's mom, and Can Cal, who's a postdoc in my lab, worked on this, is now a full professor at the University of Maryland. And there you see pictures of kids with progeria. This is a place that's different than your circumstance with paroxysmal disorders. Kids with progeria all look extremely similar to each other. You don't have the same kind of heterogeneity that you all have, and you've described so nicely here this evening by your case examples. So these kids are quite similar, but they follow a similar track, which means that in general, they're facing a very short lifetime. So insights, I'm going to go down a few of these, and then there's time at the end for you to pose questions, which I'm happy to wrestle with. So first of all, if you want to figure out what is the underlying cause, if you really want to come up with a treatment, your ability to be successful is going to be a lot better if you have an idea of the molecular pathway that you're going after. For progeria, it was possible to discover what the mutation was. I'll show you a diagram of it in a minute. And we, as many of you have done, took advantage then that we landed in a molecular pathway that people had been studying, not knowing it was going to be relevant to a particular disease, but just studying it because it was good basic biology. The mutation in progeria is almost always a single letter that normally is a C, and it's been changed to a T, and it's an exon 11 of a gene called lamin A. Exons are the parts of the code for protein. But when we first saw this, we were a bit mystified because it looked as if this might not be a mutation that would cause a problem. Because if you learned about the genetic code, It takes 3 letters in the RNA to code for one amino acid. And this change, which goes from a GGC to a GGT, still codes for glycine. And you would have therefore called that a silent mutation, and you would have walked past it. But that couldn't be the explanation, because 90% of these kids had that very same mutation. And interestingly, their parents did not. So this is a different kind of inheritance than what we heard about from Doug and Mindy. This is a circumstance where there is a de novo, spontaneous change in the DNA sequence, it being passed from parent to child in that very single nucleotide. And that turning up in multiple kids with the disease in just that letter, you have to say on a genetic basis, there's a cause and effect going on here. So what is going on? That should be a silent mutation, but it's obviously not silent. Well, again, I'm going to get a little bit into the molecular biology nitty-gritty here. It turns out that while it doesn't change the amino acid, it actually creates a signal for the splicing apparatus that is responsible for putting exons together. And so that mutation creates in the middle of exon 11, which is supposed to be continuously translated into protein, a splice donor. So if you look at the diagram there with those yellow diagonal lines, basically this causes the last part of exon 11 to be skipped. Now that doesn't seem like such a bad thing, but it turns out it is a bad thing, because that means you end up with a protein that's missing the coding potential of that last part of exon 11, which happens to be 50 amino acids. So this mutation causes you to make a protein from this otherwise normal gene called lamin A that is missing an internal piece. And that turns out to be a toxic protein for reasons that we now understand. And the molecular biology was pretty easy to figure out here because people had worked on lamin A for a long time. Again, not to dwell too heavily on the details, but you see those scissors there on the left? Normally this protein gets processed, and the last step in processing is that it cleaves off a little bit of the protein that's at the very far end. And it needs to cleave that off in order for the protein to end up in its proper location in the nuclear scaffold, just inside the nuclear membrane. That 50 amino acids that is missing when you have this splice problem contains the recognition signal for the scissors. So the scissors don't work. And so you end up with that protein in the lower right, which is the toxic protein. It still has a tail on it that's a lipid. That lipid gets stuck in the nuclear membrane. Other proteins stick to it as well. And it disrupts the cell in a fashion that, especially when it tries to go through multiple cell divisions, ultimately it can't do it. And the cell prematurely senesces, and the consequence of that is that the whole body of the child is also prematurely senescent. This was an unprecedented mechanism, but there it was. And this was possible to figure out pretty quickly because people had worked on this protein, so we went to school on it. So that had a mechanism, but okay. I'm working at this point as a collaborator with Leslie Gordon, whose son, Sam, is getting older, and all these other kids have been identified with this condition. Time is of the essence. How do we work closely and quickly to try to figure out what kind of intervention might work? I know that must be the same kind of sense of time that you all have looking at your own kids as time passes. So certainly, and this meeting is a great example of that, the prime need here is to work closely with families, and also to be sure you understand what is the natural history of this condition. If you're going to try to develop an intervention, you have to have some idea whether it works, which means you have to know what would have happened if you hadn't intervened. And that's not so easy sometimes with conditions that are rare. And so every kind of information needs to be collected to identify what would be the likely course if nothing was done. And then this gets to the point of deciding what would be, if you were going to run a clinical trial, if you had an intervention that was ready to go, what would you use as the endpoint? What would you use to tell you it worked? Or what would you use to also say, this one isn't working, we need to try something else? The FDA will require you to decide that before you do the study. It's not fair to decide afterwards, well, we measured 20 things, and one of them looks like it got a little better, so that's called a successful trial. That is a way, actually, to mislead yourself. So you have to figure out, what's my endpoint going to be? For progeria, this was pretty tough. Very rare disease. The main problem is cardiovascular, but it's not so easy to non-invasively see what's happening in the cardiovascular system. And you don't want to wait 10 years to figure out whether you've actually prevented heart attacks in a lot of kids who you just started on the therapy at age 2. So we struggled with that. I will tell you what we did was, well, first we ran a natural history study to try to identify as much as we could from kids who were available, what were the features that happened at certain ages. And so in this case, at the NIH Clinical Center, with only 15 children, because that's what we could find at the time, collected that information. It didn't give an easy answer as to what the clinical measure of intervention success might be. And ultimately, what we looked at was actually the growth curves of these kids. Because it turns out that they pretty much stop growing by about age 3 or 4. Their weights just don't go up after that point. And so if you had a therapy that was working, you would think you would see that growth curve start to go up again. And so effectively, we talked a little bit about this briefly at the dinner table, each kid became their own historical control for deciding whether the therapy was working. Because if the growth curve looked like this, and then you applied some intervention and it went like that, that would be a pretty clear sign that something was happening. The downside of that was that probably that was not the thing you were most interested in trying to treat. You were really interested in trying to treat the cardiovascular disease, and we didn't have, at the time the studies were started, a good way to do it. Well, we also had to, if we were serious about trying some therapy, we don't want to go straight to a patient with a therapy that might actually be toxic. You want to get some kind of evidence about whether it's working in a simpler system. And those are often cell culture. I know that's going on with paroxysmal disorders. If you had a therapeutic, and if you had a way of distinguishing whether a cell growing in a Petri dish was looking more normal after you added that kind of candidate drug, that would be a good sign that we're on the right track. But that means you need to have some way of distinguishing whether that cell from a condition is different than a cell from somebody who doesn't. So for us, we were perhaps in an easier place than some conditions because once we knew what to look for, skin cells from kids with progeria are actually quite visibly different than normal cells. Particularly so if you let those cells grow in culture for a while, so they divided, because every time the cell divides, this toxic protein makes things a little worse and makes it harder and harder for the cell to be normal. So what you're looking at in the top there is a single cell. When you're looking at the rim around the nucleus, which is where this lamin A protein is, you're looking at the lamin A protein. On the left, that's only 6 passages growing in culture, and then it gets to 13 passages, and it's starting to look kind of lumpy, bumpy, and you get to 26 passages, and that's a very abnormal looking nucleus. So that was our measure. If we were to come up with a treatment, we ought to be able to do something about the lumpy, bumpy nucleus if we added a drug. Okay, now, what are we going to do in terms of coming up with a therapy that would build upon the molecular understanding we had of the disease? Again, I think what we're trying to do is to reduce the amount of that protein that you see at the bottom there. That's the toxic protein that has that lipid tail that gets stuck in the membrane. We call that progerin, and progerin is the abnormal form of lamin A. We would like to reduce or eliminate that. Well, what could you do? Well, how do you make that? Actually, there's 2 steps to get there. And the first one is to add that lipid tail. And that lipid tail is called a farnesyl group. And so the enzyme that does that is called farnesyl transferase. Suppose you could block that enzyme, and then you'd be dialing down the amount of the toxic protein. Maybe that would be a good thing. You might immediately say, wait a minute, what other proteins are also modified by farnesyl transferase? And that would be a good question. And the answer would be, well, a couple hundred of them, actually. So you're kind of mucking around with biology here by trying this out. But we already knew that drugs had been developed, actually with the intention of treating cancer, that are farnesyl transferases. Companies had done this 20 years earlier because they thought it was going to cure cancer, and it didn't work. But we could get our hands on those compounds. We did know that they'd been given to human beings, and they didn't seem particularly toxic. So the idea that you're really messing up biology apparently hadn't happened. So we got our hands on one of those. And here you see just single cells, two different patients. On the left with no treatment, in the middle at 1 micromolar, on the right with 2 micromolar. And what we could see in just 72 hours is those lumpy, bumpy nuclei got much prettier, that their shape got corrected. And that was actually pretty encouraging. Didn't actually expect that in 72 hours you could see that kind of response for therapy. And that was very reproducible over and over. But you know, these are cells growing like petri dishes. So would you want to come up with some other model that's closer to a patient without putting a patient at risk? Well, what we often do at that point is to make a mouse model, and make a mouse that has similar problems and similar molecular basis for those problems, and then see whether our proposed treatment would help them. So, mouse model time. How do you get about setting that in place? Well, there are various ways to do this. I won't advocate that this was the right one. It was the one we used now quite a few years ago. We basically wanted to put into the mouse that same mutation, and we actually put it in the human gene, because we weren't sure whether the mouse gene would behave the same way. So a big piece of DNA here, 164,400 base pairs, got inserted into the mouse germline. That is, it could be passed from parent to child to grandchild. And we had already retrofitted that particular piece of DNA with the Progeria mutation, which is called G608G. There's some other genes along for the ride here, and you got to worry a little bit about whether that's causing trouble. We were able to show that those didn't seem to do much. So then we could make a mouse that had the Progeria mutation, but it didn't have anything that looked like the same condition. Well, turns out it's not too bad a model. And because you put this into the mouse chromosome, you can actually breed 2 of those mice together that carry it and get the double copy. And then, as you might expect, the mouse is a little more severe. So what you're seeing here on the left, a so-called wild-type mouse, a normal mouse. In the middle, a mouse with 1 copy. On the right, a mouse that has 2 copies and is more severe. And you can see, particularly the one that's 2 copies, it's smaller. That is the growth retardation. There is something going on with the spine. There's a kyphoscoliosis, which reflects to a certain degree the skeletal problems with progeria. There are also joint problems. And as they get a little older, they also start losing their hair. So it's not too bad. But the big question was, what about the cardiovascular disease? Because that's the thing that we know is going to be most serious. Does the mouse have that? Because if it does, we're in a better position to use this to test the therapy. It turns out it does. So here you're looking at the aorta, that pink swirling thing. The wild type one, it's hard for me to point this out without standing next to it, but I hope you can see there are a lot of cells in that wild type aorta. If you look at the double copy mouse, right in here, there should be cells in there, and there aren't any. Basically, the cells that are in the middle layer of a large artery like the aorta are the ones that take the worst hit from this toxic protein. And by the time you get a mouse at this age, which is 6 months, those cells are pretty much all gone. And that then leads to cardiovascular disease. So it turns out this is a pretty good model for cardiovascular. So now, okay, we've got a chance to try this therapeutic, not just in cells, but in an animal. This is maybe an easier picture to see what happens in the Progeria mouse. At the top, you can see a wild-type mouse. What's called the lumen there is where the blood flow normally would happen in that aorta. The stain there in green is for a protein that's in those cells in the middle layer, those so-called vascular smooth muscle cells. That's what it's supposed to look like. In the bottom images, though, that's progeria. Those cells are all gone. Now, this is a single copy mouse, and this is at 9 months. Then I'll show you what happens when you treat that mouse, starting at time of weaning, with this farnesyltransferase inhibitor, the same drug that looked like it was helping cells in culture. And it was pretty dramatic. So you can even wait a little while and let the mouse get a little bit of cardiovascular disease, treat with the drug, and it seems like it's somewhat reversible. So now we're excited, and now we think, okay, maybe this is sufficient to move this forward in a clinical trial. I just want to emphasize, though, how important it is to be sure that you have that kind of supported evidence before you go to a phase 1 or phase 2 trial with actual children, because you don't want to do something that has toxicity that you could have anticipated if you have a chance to learn it first. And the FDA generally won't let you unless they're convinced that you have done that kind of work to be sure that the benefits are sufficient and the risks are going to be as best understood as you can be tolerable. So, with those options, you also need partners. MCATS has already been mentioned, has been part of the conversation here today, I understand, through the Rare Disease Clinical Research Network, through their ability to provide high-throughput screening to try to find potential small molecule therapies for disorders like the peroxisomal disorders. They are one of the most critical parts of NIH for assisting in the search for a rare disease solution, which is why I'm really glad that you all have linked up so well with them. And Joe has been a good part of the glue to make that happen. And the best thing you can do is to put a postdoc at MCATS who works on peroxisomal disorders, because then you've got somebody who's thinking about it every day, and you've managed to get all of the staff there to think about it every day too. It's a multiplier effect. Postdocs have a return on investment that's a lot greater than most people realize when you set them in this kind of position. And again, MCATS offers you a lot of things that are pretty important to be able to do, and that includes certainly the Rare Disease Clinical Research Network, but also the Therapeutics for Rare Diseases program, TREND, which is specifically designed to encourage the development of treatments for disorders that are not going to be pursued right away by companies because they seem too risky. And basically what MCATS aims to do is to help you move from the left side of this diagram to the right side, all the way from having an idea about a target to what is ultimately an FDA-approved therapy. A lot of other boxes on here which are resources that you can tap into in that case. So I'm really glad that you have that connection, and they're going to, I think, be quite helpful as you've seen try to negotiate what is admittedly a very challenging pathway to go from left to right. For Progeria, in order to move this from animal studies to human trials, the ultimate decision was that the trials would be run in Boston, where the Progeria Research Foundation has strong connections with clinicians, and particularly clinicians at Boston Children's Hospital. And that was done in part because of the idea that the drug they wanted to try, lonafarnib, which is a farnesyltransferase inhibitor, had already had experience in the hands of the docs at Boston Children's because they had been trying it for other kinds of childhood cancer. So they already knew how to use this drug, what its potential toxicities were, how to manage those. And that turned out to be pretty helpful, having clinicians who not only wanted to do something for the disease, but even knew something specific about the therapy that was being tried. This looks rather daunting with all the names that are here that ultimately have become involved in this over the course now of about 8 or 9 years of clinical trial efforts, and not all of them are engaged at any given moment. But maybe just as with this condition, this is kind of a magnet for people who want to try to do something in a circumstance where you have children that need help. And so all these people, most of them just volunteering their time, got engaged in this same enterprise. And again, with a tight connection with the Progeria Research Foundation, which was started those many years ago by Leslie Gordon and Scott Burns. And in order to be able to run the trial, you have to figure out how are you going to enroll participants. So you need to be quite sure you have the diagnosis correct. So the trial needs to have access to genetic diagnostics that are what we call CLIA approved. For a disease like progeria, where there aren't enough patients in the United States, there's a great deal of effort that went into actually figuring out how to bring patients from other countries with travel visas and passports and so on. In this case, because the drug lonafarnib was made by Merck, but had never actually been approved for use for any disorder, remember it had failed as a cancer drug, Merck still had a supply of the drug. If we had to pay for it, it would have been pretty prohibitive, having convinced them to donate the drug, which they did, and much credit to them for that purpose. And then there are all the regulatory issues. If you're going to actually propose this for human use, you have to convince the FDA that you have sufficient data. so that they will give you what's called an IND, an investigational new drug approval. You have to convene your institutional review board, your IRB, and tell them what you're proposing to do, and they have to deliberate about whether they think this is ethical in terms of benefits and risks. And there's hospital reviews of other sorts. If you're going to be running a clinical trial, you may need a data safety monitoring board that is not blinded to what's happening. If it's a blinded study, they're keeping track to make sure that you're not encountering problems that you aren't aware of. All of that needs to be thought about as you move into the direction of setting up such a trial. And I don't mean to make this sound daunting. This is all manageable, but you need to kind of plan for it, think it through, make sure that you have the appropriate experts to guide you. Again, MCATS and other parts of NIH, like the Rare Disease Clinical Research Network, can help you a lot with this. So the farnesyl transferase trial was in fact initiated, and it involved just 28 kids, because that's all we could find, and it ran over 4 years. And at the end of this, it was possible to publish the results. And as I told you, the original endpoint, which was weight gain, was in fact met, but just barely. And it was pretty clear that some kids had weight gain that was reasonably impressive, and others didn't have much at all. But if you look across the whole 28 group, you could say, yes, we do seem to have benefit. But again, that wasn't what we were really looking for as far as the cardiovascular disease. So in the course of the trial, it was also possible to come up with a better measure of cardiovascular disease, and that's what I'm showing you here. This basically measures the stiffness of a blood vessel. Remember, those large arteries are the big problem here. Is there a way to assess non-invasively what's going on there? And it turned out there was, but it kind of got invented in the course of the trial. The gray zone is where kids would normally be. All of the kids before therapy had a much higher level of stiffness of their arteries. And during the course of the treatment, all but one of them showed a drop in that, and that's highly statistically significant. So when we saw that data, even though, let's be clear, this was not the primary endpoint that we had originally designed, this did convince us that something good was happening. Now, all the kids that were on this trial have been on this drug ever since, because this looks promising. So we even now have a bit of evidence about what it's doing to survival. And this is what's called a Kaplan Meier curve. And the solid line is historical controls. This is what survival has looked like for progeria. And you can see that drop-off, which happens at about age 10 or 11. And very few, if any, kids are living past about age 16 or 17 without treatment. Whereas with the dotted line, that is the cohort that's being treated with lonafarnib. And it is highly significant that we have extended their lifespan. But it's also, tragically, still the case that virtually all of these kids seem to hit about age 20, and that's as far as they can go. So what does that say? That says we had sort of a success, but it's not the success you were ultimately looking for. And that often happens, whether you're talking about a common disease or a rare disease. The first drugs for HIV/AIDS, it was a big celebration because they actually seemed to have some benefit, but they were not sufficient. You have to keep pushing to the next generation and the one after that. So that's where we are now at the moment with progeria. And there's been at least one instance where things didn't go the way we had hoped. That first line there, lonafarnib with 28 cases, that's the one I told you about. The next trial added 2 other drugs to lonafarnib. One a statin drug, and the other one that was supposed to strengthen the bones, because it's a bisphosphonate. And I won't show you the data, but the indications are that that actually made kids worse, that it resulted in calcifications in the skin and potentially in large arteries. And so that trial was stopped, despite the fact that in the mouse model, this looked like it was going to be a good idea. In humans, it was not. So now everybody's going back to lonafarnib, and we're just engaging in the last year with another idea. And this is a totally different approach, a different drug that does a different thing that maybe would be synergistic with lonafarnib, something called everolimus. And I don't have time, and you don't probably want me to have time to explain what the mechanism is that leads us to believe that this might help. But again, this shows significant benefit in cell culture. That's what that little picture is. And it also shows benefit in mice. And it's a drug that's been used. This one's actually approved by the FDA for other purposes. It's used for treating kids who've had an organ transplant. It's a mild immunosuppressant. So we know what its toxicities are, and FDA decided to allow its use in this circumstance. And we're up to 17 patients that have been treated with both drugs. I can't tell you anything at this point about whether that's adding benefit or not. It's way too early to tell. But when you look at the list, because a lot of people have been thinking about this, of things that might be tried, this is also a bit daunting. If you only have maybe 100 patients in the world available for clinical trials, we're not going to be able to run all of these. You have to really figure out what's your priority. The first two we talked about, the farnesyltransferase inhibitors and that everolimus, which is an mTOR inhibitor. Actually, one that we are now deeply engaged in pursuing is that third one. And this is one in collaboration with a company called Sarepta, the same company that has FDA approval for treatment of Duchenne muscular dystrophy with a similar strategy. And this is actually a direct effort to block that abnormal splice that I told you about, not to change the protein downstream, but to change the actual initial problem of that splice that shouldn't be there. And this, I have to tell you, looks very good in cell culture and looking pretty good in mice as of last week as well. But what are we going to do here? We're already engaged in a trial that's involving a fair number of the kids with this condition that we want to see through. How do we start another one? And this will happen, I think, with lots of rare diseases, just something to think about. And then there's this other long list of potential interventions, which are at various stages of being evaluated, many of them quite early. A couple of final points. Translational research of this sort may yield basic information that is quite relevant to other things. Think about this when you're trying to figure out how to get grant support. If you can show that stuff you're learning about a rare disease has an impact on common disease, that's a good thing. That turns out to be true. All of you are making progeria. That abnormal splice, well, you're using it a little bit too, without a mutation even. And so this probably is a significant player in the normal process of aging, something that we all are afflicted by at various levels. And believe me, we made every possible big deal out of that to try to get funding for this extremely rare disease, arguing that it means we're going to learn about something that congressmen care about too. So seek out those kinds of opportunities. We're all in the same boat with it. And then finally, never forget the reason for doing all this work. I started with Meg, Casey, and Sam. Well, here's Sam, a little bit more grown up. Sam with Cookie Monster and myself at an event about 3 years ago. Sam, who was the star of the show in a HBO special called Life According to Sam. And Sam did a TEDx presentation, which was also pretty spectacular. Sam, who I'm sorry to say we lost 2 years ago at the age of 17, had his philosophy for a happy life, which is a pretty good set of recommendations. You can read what they are there, and especially never miss a party if you can help it. The last one. And I know this organization by being here in the evening in a nice restaurant is trying to live up to that as well. So I hope this is useful in terms of the parallels that exist here. Again, there are some very significant differences as well. But as you think about the path that this set of disorders, the paroxysmal disorders are on, maybe there are some bits of wisdom here that might be helpful in the planning. And I just want to say for myself, as the director of NIH, certainly representing the thousands of scientists that were in that environment, this is our dream too. That we can take that diagram that has 6,000 diseases we can diagnose and turn that into 6,000 diseases that we can treat. That's going to be a very significantly challenging effort, and it may take a lot of groups like this to roll up their sleeves, find the right resources, and find the right smart scientists to work. I think you're very well down that road, and my hopes and dreams are very much with you, and my willingness to assist in any way that I can. So thank you all very much for listening. So while you're thinking about a question, I want to introduce my wife, Diane Baker, who came in a little on the late side for a whole Certainly, rare disease is something that we work on together. Diane is a genetic counselor, and we've had many opportunities to meet with groups like this and hope to be helpful.
All right, so we're going to take a couple of questions.
Yeah, because I know I've been on a long time.
We appreciate it. Who's got a question?
There's one over there.
So, in thinking about this common glycine to glycine mutation that happens in 90% of the cases, I guess I'm just thinking if there's a mechanism as to why that site specifically is being spontaneously mutated. Why is that so sensitive?
Yeah. So, great question. First of all, it should be recognized that we all have new mutations in our genomes. We even have a pretty good idea of how many. Somewhere between 60 and 100 mutations that were not present in your parents that you have. They particularly happen in nucleotide sequences that are C followed by a G, because that is a situation where the C is often methylated, and a methylated C gets looked at and called a T. And that's exactly what this is. That C to T change is one of those hot spots for mutations. But still, it's pretty astounding that out of 3 billion base pairs, that particular one gets spontaneously mutated often enough that we recognize a disorder as a result. There's another example of that, which is achondroplasia, where it's also, in many cases, but not all, a de novo mutation. It's the same thing, it's a CPG in the middle of an exon that results in that case, in this sense, with the protein, not a splice problem. But yeah, it's sort of astounding. Sometimes genetics works at all. There are all these things that can go wrong so easily, but that is in fact the basic explanation that we've come to.
Thanks, wonderful talk. My question is, with so many disorders that so desperately need treatment, how do you make yourself stand out for special attention?
Great question. And it's basically a combination of demonstrating a scientific opportunity, because if NIH, for instance, is trying to make a decision about where to make investments in limited dollars, you want to have some confidence that they're going to make progress. So being able to have a path forward that's based upon a good molecular hypothesis is a really good start. Certainly demonstrating need and to have a clearly compelling circumstance, and childhood diseases tend to be particularly compelling, and to make the case that this is both scientifically needed and in human interest potentially going to be valuable in a major way. That's your best chance to sort of get up there and get some attention. It helps to have a community, that it's clear that this is not just an isolated idea of an investigator somewhere, but they're also linked up with other expertise, so that if the snag gets encountered, or there's a problem, or you're trying to figure out how do I turn this into an animal model, or how do I develop a high throughput screen for the MCATS robot, that it's clear you have sufficient number of disciplines represented that you're going to be able to do that. And that's clearly one of the things that's happening here. Putting that all together, so if you imagine yourself as trying to look across 30 or 40 different disease applications, what would make you go, wow, that one looks like it's poised to go somewhere? Put all those features together and you've got the best chance. And then make sure Joe is excited about it.
I've got one right here. Hand it to Steve Craig.
So, Steve, you're from UNC. I was going to ask you about this community, Progeria community. A lot of these very rare disease communities are kind of, they're struggling with identifying that first lead candidate, doing phase 1 trials, maybe doing phase 2 trials. When it gets to the point of actually wanting to have a drug that's successful, that's can be prescribed by doctors, the model is to de-risk something and then have a company take it across the finish line. But with a disease like this, I'm not sure that there is a business model. I'm not sure that there is something for a company to step in like that. So is there another plan for these ultra rare diseases to get it, once we have something that works, to actually get it to where it can be readily available to all the patients?
Yeah, that's a very challenging question. You may notice here with Progeria, the two drugs that I talked about are actually drugs that already had a lot of work done on them by companies. So some of that investment was made not because of this disease, but because of others. And if you can figure out a way to repurpose a drug that was developed for some other application and show that it works for your rare disease, that's a fantastic opportunity. And I would say, for instance, that if you had an assay you're pretty convinced is good at detecting potential small molecules that would be a benefit, the first thing you want to do is to screen that against all approved drugs. Because if you get a hit there, you've just leapt about 8 years and $500 million ahead in terms of the work that has to be done to get that to a successful outcome. You don't always get so lucky. I don't know. It is certainly the case that companies that used to say rare diseases are not attractive, you can see more and more. It's maybe not getting quite down to the level of, well, we only have a few dozen patients available, but if it's a few hundred, it's starting to look like it might be enough for a company to invest in. And here's where the government, I think, ought to be able to try to step in if there's absolutely no other opportunity. And that's what TREND is all about, the Therapeutics for Rare and Neglected Diseases. And they have carried things, certainly to phase 2 trials, where there didn't seem to be an opportunity. Even Pixie recently kind of followed that rule, and then it did actually get out licensed. So other than those sort of opportunities of trying to get lucky and have a drug that's already been developed for some other purpose, or convince a company that even though it's rare, there's an investment here because you've de-risked it enough, the government is your third opportunity. That means the government needs to be well funded. Oh, did I say that? There's a question in front.
Hi. How do you think that physicians or any type of caregiver can best provide support for patients who may be suffering for a rare condition for which there is no promising therapy?
So I think in reality, and this has been well demonstrated by the stories we heard this evening from Melissa and from Doug and Mindy, is that physicians often end up knowing less about the disease than the families do. And especially that's true now with internet and the ability to connect families to each other. And that's one thing, as we heard in the examples, that has made a huge difference. That wasn't true 20 years ago. So physicians ought to be in a position of saying, look, I don't know enough about this to be your source. I'm going to encourage you and help you to link up with other families that are going through the same story, and I want you to teach me. I'm not just going to teach you. I guess the other thing, and I love the way that Melissa told this story, is physicians can help those families realize that, okay, this is not a case of disease X. This is your child. This is Johnny or Glenda. And your goal here is to make it possible for that child to have a happy life and not just to be considered an example of a disorder. To not over-therapize, I think that was your word, the particular approach, because you're still wanting to create an environment where that child has an experience of interacting with other kids, interacting with mom and dad, everything that you can make as normal as possible and not deprive them of that opportunity. And that's something I think you heard from a physician. Physicians aren't always so good. Melissa, I think we've worn them out.
Okay, gosh, I know, it's late. Okay, well, let's thank Dr. Collins one more time for being here.